Hi, it's a great pleasure to have this opportunity to share with you some of our findings on co-regulator HIC5. These have been published recently in Molecular Endocrinology. HIC5 has been shown to bind several steroid receptors, including glucocorticoid receptor and androgen receptor, and it binds some other transcription factors as well. It is a very versatile protein and acts through multiple mechanisms of action in regulating the transcription of target genes of these steroid receptors. Previously, we characterized the global co-regulator function of HIC5 in the context of glucocorticoid receptor alpha signaling. In this work, we have extended our investigation to include two other steroid receptors, the glucocorticoid receptor gamma and the estrogen receptor alpha. In this work, we have shown that HIC5 has a selective co-regulator function for modulating the transcription of several steroid receptor target genes. We have also shown that HIC5 can restrict the genomic occupancy of these steroid receptors on a subset of genes, thereby preventing their regulation by hormone. In our previous work with respect to glucocorticoid receptor alpha signaling, we showed that HIC5 regulates distinct classes of genes that are responsive to hormone. The HIC5 modulated genes were those that were regulated by hormone in the presence of HIC5, but their transcriptional activation or repression by hormone was significantly affected when HIC5 was depleted. We also found another subset of genes which we call the HIC5 block genes. These were transcriptionally not regulated when HIC5 was present. However, when you get rid of HIC5, these genes become significantly upregulated or downregulated by glucocorticoid. On these genes, HIC5 maintained a closed chromatin state and thereby precluded the efficient recruitment of GR to its DNA regulatory sites. Here, in a genome-wide analysis, we looked at the effects of HIC5 depletion on overall genomic occupancy of GR-alpha, which is the major isoform of GR, and we did this by ChIP-seq analysis. We found that HIC5 depletion dramatically increased the global number of sites occupied by GR-alpha. This number almost doubled in cells that were depleted of HIC5 compared to those that had HIC5. This included the appearance of many new peaks, which were called the HIC5 blocked peaks, appearing only upon HIC5 depletion. We also analyzed by differential MA-norm analysis and found that there was a significant gain in the overall GR-alpha binding to DNA when there were reduced levels of HIC5. The events that were shared in the two datasets, a higher percentage of them had stronger binding in cells where HIC5 was depleted. We further correlated the overall changes in genomic occupancy of ligand-activated GR with the DEX-regulated status of genes. And we found that many blocked peaks were associated with the HIC5 blocked genes. These blocked genes were not just unique to the GR-alpha cells. We found, similar to GR-alpha, a subset of HIC5 blocked genes that were in cells that were stably expressing either GR-gamma or ER-alpha. Many genes remained unregulated by their respective ligand in these cell types when HIC5 was present. However, when we depleted HIC5, these genes now became robustly activated by ligand. In the GR-gamma and GR-alpha cells too, similar to our observations on GR-alpha, we found that HIC5 blocked target gene regulation by preventing the occupancy of both GR-gamma and ER-alpha in response to ligand to their DNA regulatory sites. Our results presented here strongly suggest that HIC5 is an important factor that determines the transcriptional status of several genes. Because HIC5 expression is highly tissue-specific and DNA binding sites can differ between different cell types, we think that by understanding the molecular mechanism through which HIC5 precludes the hormonal regulation of genes will provide additional clues into understanding how selective transcription factor binding is achieved in different cells. I invite you all to read more details about our work in the article published in Molecular Endocrinology, and thank you very much for watching.